Hello. Welcome back to my channel if you were here in the first place. Um, I started this channel 2 years ago over the summer because um, people left for summer break and I was finding it really difficult to focus when I didn't have peers around me who are doing the same thing. So I started this channel to give myself a fake sense of community. I had this plan to post videos and engage with, people. And as you see, I've only posted one video that summer, so the plan failed. And fast forward, it's been 2 years. I very much let this channel slide. But now I find myself back in that situation where it's summer break and all of my peers are doing something else, but I'm trying to be motivated. Um, so I'm gonna try to do that again and hopefully make it interesting, for myself and for my viewers. But honestly, to like, there's no one's gonna see this except maybe myself in 10 years, in which case, hi. Um, so the idea is I read a lot of papers. I enjoy reading papers. It's why I enjoy doing research. And this summer, we're just going to try to read as many papers as possible related to my project. So that as the semester starts, I could get the experiments going and whatever. The problem with that is reading papers without doing experiments is really, really boring and you get burnt out really fast. Um, that which is why I started the channel. We've been over this. I will keep this channel really low key because I've been listening to a lot of podcasts, for better or for worse. I may have Listen, I've seen too many middle-aged men find a good podcast and lose themselves over it. I may be following that path, but my takeaway from podcasts as of now is I'm gonna keep this channel low key. So I would yap like this. Um, we'll eventually get into the science. Actually, let's get into the science because I feel like the yapping is done. Anyway, welcome. I'm I present interesting papers that I find. I did not write it. I am in no way related to these people. I found them on Google Scholar. Um, I really enjoy the papers that I present, and like if in some magical world that any of the authors of these papers come across these videos, like I have so much respect for your work, and the reason why I share is because I aspire to become you one day. And then another disclaimer is that because I am in no way involved in the research that goes into writing this paper, I may get stuff wrong. Um, if I do, let me know. I am sorry. I expect to get things wrong because there's just no way that I have the ability to understand things on this level because this is such a beautiful, this paper, for example, it is such a beautiful piece of work. I understand exactly why they did each experiment after the other, which is my criteria for having for writing a good paper is that you told a logically coherent story, and then you're just maybe a few seconds lagging behind the logical process of the reader. So as your readers asking these questions organically in their minds, you just sort of follow along that logic with your narration, and your readers are very satisfied because all of their questions get answered at the end of the day. Anyway, that's the yapping part. I also have a cat in my house. So if you see a furry creature behind me, in front of me, don't be alarmed. Anyway, let's get into it. This is a paper published, I think, a month ago on Physical Review E. It's called Nuclear Pore Complex Distribution on the Nuclear Envelope, Insights into Curvature, Chromatin, and Actin Contributions. I think I'm gonna once again, this is low key. I don't have a script. I just pulled up this paper, I read it, and now I'm making a video. So follow me along. I think I need to start with a rough biological background just in case we have hardcore physicists in the chat, which again, why would you watch this video? It's not a good video. Do something better with your life. Anyway, um so how, so I am interested in nuclear mechanobiology, which is why I came across this paper. The idea is that mechanical forces can deform the nucleus of a cell, in some way that impacts things like stem cells and like cancer cells and other things. It has a lot of applications, but basically it's a relatively new idea that like all sorts of mechanical signals can impact the nucleus. Before this point, people were more focused on the biochemical side of things, of how like I guess signaling processes work in the nucleus. And I feel like from my perspective at least, again, I may be wrong. The mechanobiology part is lagging behind the biochemistry part, because we don't really understand like compressing the nucleus or like osmotic pressure or like hypertonic shock within osmotic pressure are things that we do to cells. And it is relevant because stem cells are able to sense the stiffness of the environment they're in and differentiate based on the stiffness of the substrates that they're on. And it's an interesting phenomenon that we don't really understand. And one of the many hypotheses is that the nucleus, being the most stiff part of the cell, is responsible for sensing the stiffness of its environment through something called a cytoskeletal what is it? It's a comp like a cytoskeletal network that connects the nucleus to the membrane. I think this is where like a lot of like material transport happens within the cell. Like whenever like a thing needs to go from this part of the cell to another part of the cell, it would be enclosed in a little bubble and then travel along the cytoskeletal network to get where it is going. Again, I could be wrong. Um, and then and then that's an example of why stem cell biologists are interested in nuclear mechanobiology. And then cancer biologists are interested in mechanobiology because cancer cells sometimes, well, cancer cells definitely need to squeeze through very tight like openings in order to propagate to other parts of the body. And sometimes like these like tiny openings are like smaller than the nucleus itself. So the nucleus needs to like deform itself somehow to get the entire cell through. And I think okay, I'm gonna stop saying I could be wrong because that's just the baseline assumption of everything that I say. Um, actually, let me make sure I'm actually recording. I am actually recording. Okay, that's good to know. I was scared for a second because I didn't see a recording icon on my screen. Anyway, cancer biologists are interested in nuclear mechanics because they don't know why cells manage to squeeze through teeny tiny spaces when the nucleus is so stiff. Anyway, I feel like that's enough background on why I'm interested in nuclear mechanobiology, and we're gonna get into this paper. Um, I should probably start with the abstract. The idea is that, okay, the idea, actually, we're gonna skip the abstract because like explaining the abstract is like the entire paper basically. I would much rather like follow their logic along what they did instead of just tell you everything based on what I remember. Whatever. So as you see, I have a lot of annotations. Uh, um, in I'm just this is not important, but like underlined in purple are things that I did not know before reading this paper that I had to look up in Google or whatever. It's just a nice thing to keep track of. Like when I started reading about biology stuff, I had to look up like, oh, what's the cytoskeleton network? Like, and now I just know it. So, ignore the underlined blue things. Sorry, the underlined purple things. In blue, I have background information and or experimental methods. For example, like it is important for us to know that the nuclear pore complex is not very mobile on the nucleus. And then later on, you would see highlights in green, and those are results, like from the authors. Um, and I think that's all the annotation methods that I have. Anyway, the nuclear pore complex is basically how the nucleus communicates with the outside. Like if signals needs to come from the cytosol into the nucleus, it goes through the nuclear pore complex. As the name suggests, it's just a bunch of holes. These holes are being held together. Actually, hey, this is my cat. Not to like diverge attention, but hello. Anyway, what was I saying? Oh, I remember. Okay. Anyway, so as the name suggests, it is a hole. It's a pore. Around the hole, there are like very specialized proteins called nuclear porins, NUP. There are like, oh, as they tell us there are more than 30 different proteins, and that's just, like holding together the structure of the nuclear pore complex so that this hole can exist on top of the nuclear membrane. And then nuclear membrane is like the nuclear envelope is the nuclear membrane is like a double membrane. So I'm gonna use that interchangeably. Um, as we've already said, the nuclear pore complex doesn't really move around the cell. Um, but there's a hypothesis in the scientific community that the spatial distribution of nuclear pore complexes can locally modulate transport rates of like how often information comes into the nucleus and leaves the nucleus. Um, which is why the authors of this paper are interested in the spatial distribution of NPCs. Um, so Okay. What's really annoying is I know, actually, yeah, my cat is. Anyway, what's really annoying is I know what they did, but I don't know which part of the paper they write about it. So I'm just gonna narrate what I think I remember and not really go based on the paper, which actually I feel like I should have just follow the abstract. Anyway, so these researchers are interested in distribution of the nuclear pore complex. There are two ways in which the nuclear pore complex can form. One is during or right after cell division. One is somehow like during cell growth. Um, I don't think exactly how that happens is important for their cause. It's just, it's difficult. So on a regular basis, like the majority of the time, the nuclear pore complexes are very much fixed in space. Which makes like studying their spatial distribution a little easier than if they were to move around all the time. And then they were inspired to do this because the nucleus of yeast was demonstrated to have like properties that sense membrane curvature. So like curvature of yeast nucleus impacts the spatial distribution of NPCs on those nucleus. This is what that section is doing. Which is why they were like, oh, does that apply to humans as well? So the keywords of this paper is like the nucleus, NPC, spatial distribution, curvature, and oh, another thing that I probably should mention. The nucleus contains transcriptional information for the cell. Um, there are two states that a DNA can rest in. They could be euchromatin, which is like active transcribing, making proteins, doing the work. They could also be heterochromatin, which is like temporarily, I think, silenced. But they're not dead. Like they exist. They're just silenced. They're not transcribing. They're not actively producing proteins. I'm pretty sure they can be turned on with like various different methods, but like in the exact moment that we're studying, that they're not active. So one of the hypotheses that the authors of this paper have is that nuclear pore complex is more concentrated on euchromatin regions because they are like more active. Like they need more information from the outside. They need to give more information to the outside. But if you are a heterochromatin region, then you're like practically you're not doing much. So you don't need that many openings on top of you, close to you, so that information can flow in and out. And that's one of the processes that they test later on. I think that's all the background information. The cells that they studied in this paper are C2, C12 myoblast cells. I don't know whether that impacts our understanding. Okay, let's get into the results that they did. I am going to completely ignore the text and just describe the images. Okay. What they did is they had a bunch of cells and then they isolated each cell so that they get a bunch of nucleuses that they could image with super resolution microscopy, which they abbreviated into SIM. I don't know where the I comes from. It's SIM. Um, in this graph on the first row, you see one image that they took of one nucleus. Um, Basically, they want to construct a 3D image of the nucleus. So they just put the nucleus here, and then their um, microscopy technique like slices the nucleus like at various levels. I'm pretty sure they said it was like 10 nanometers in between each slice. And then on the first row of this image, you're seeing one of the many slices. So like if you slice the nucleus every 10 nanometers, like going upwards, and then you piece those slices together, then you get a 3D mold. You do have to do a little bit of interpolation to make it a smooth manifold. But this is how they created a 3D image of each nucleus. And then on the second line that says sum, you could see like these are the 2D projections of the 3D images that they generated from stacking a bunch of images like those on the first line, like on top of each other. Um, so actin is um the thing that is in the cytosol compressing the nucleus when necessary. So you could see here, this is a, oh, what did they call it? They call it a T something. Let me find it. Ah, there we go. A transmembrane actin-associated nuclear line. This is when an actin fiber like completely cuts across the top of the nucleus and indents the nucleus a little bit. And then this is one of those examples. DAPI, or that's what I'm calling it, DAPI, stains DNA. It tells you where the nucleus is. That's pretty much all it's doing. NPCs, this is nuclear pore complexes. And then merge is all three of these added together. Panel B is a side view of one nucleus. So given this example, if you cut the cell on this like direction, you would, and then look at it from like that kind of way, you would see this. And then this orange line corresponds to the transmembrane actin network line thingy, like the line that is actively compressing the nucleus. Anyway, this is just to show you what a 3D construction looks like. And then in panel C, this is the interpolated surface on which the nucleus lies. Um, what were their conclusions for this section? This is why color coding works. Now that we've explained the graph, what are the conclusions that they came up with? Ooh, I don't think I flagged any important. Oh, okay. So one thing that I really respect that they did for this paper is they're conscious that their imaging technique might have missed a few NPCs. So in order to justify that the amount of NPCs that they managed to sample is good enough for the results that they derive from it later, they manually took out, like threw away, um, a few percentages of the points that they imaged. And all of this is dumped in their supplementary figures. Like they took out 5% of the NPCs that they actually imaged, and then 10% and then 20%. And then they constructed a surface based on the remaining NPCs, and they compared that surface to the actual surface that contained all of the NPCs. And then they noticed the differences between these surfaces. And I think their conclusion here is that like even if they missed 50% of the NPCs that should exist, the surfaces that they construct is still like good enough. So I think that's very strong justification for how they constructed the surface. And honestly, it's just really neat science that they're doing that like they care about like undersampling and they're checking their logic. Anyway, again, mad respect to the authors of this paper. There's a reason why I'm presenting it. I have absolutely no criticism of this entire paper. I wish one day I could write a paper like this. Anyway, um, okay. I think that's everything I wanted to address for this graph. And then we move on to figure two, which I think is one of the most interesting parts of the paper. I'm gonna drink water before we do that. Good. I have a mug that says, it's the Loch Ness Monster, I think. And it says the most important thing is I believe in myself. Believe in yourself, everyone. Anyway, okay. Hmm. Figure two, they want to know whether NPCs are clustered or not. Um, in doing so, they use two methods. One of them is called the Ripley's K function. The idea is, hmm, ooh, okay, hold on. I don't actually remember what the K function is. Like, I don't remember the mathematical formula. I didn't really go into it. That's a problem. Okay. I'll look into that after making this video. But for now, um, the idea is if a bunch of points are randomly distributed on a surface, then the K value should be equal to pi r squared. If the K value is lower than that, it means that the points are further away from each other. If the K value is higher than that, it means the points are clustered together. This is a little diagram. Um, on the first panel, this is a low K value. On the second panel, it is like a zero K value. And then on the third diagram, this is a positive K value. Um, this is usually done on a two-dimensional surface, but in our case, we have a three-dimensional like a 2D surface sitting in three-dimensional space, which means that instead of using the Euler distance between points, we should have been using geodesics. But it is very difficult to compute the geodesics and of like, I don't even know how many points are on this thing, like 800 points. So instead of doing that computationally expensive task, they decided to simulate through Monte Carlo, I think, a surface with NPCs that are like just randomly distributed. That's what they mean by simulation CSR. And then they projected this surface onto the XY plane. Um, and then they sampled it. Okay, I should have explained R. That's not good. Okay. We're gonna skip that. I can't explain it, but I feel like it's not necessary. Basically, this blue line is like what would happen if um NPCs are completely randomly distributed on the surface. And then the yellow is like what we actually got from the nucleuses. And as you see at lower values of R, um, our experimental value is lower, which means that the points are like less clustered. And then at higher values, like it's higher, so like the points are more clustered. R is basically a variable that can be put into the K function. Um, it's so the K function, I think, is defined something like how many points can you find within radius R of each point on the surface. Um, which is why when you have a lower value of R, it is less likely for you to find neighbors. In fact, when your R value is lower than the radius of each NPC, it is impossible for you to find another NPC because, they don't overlap, which is why our both of our curves tend to zero, like as R is going to zero because that. And then when you have a larger value of R, it is more likely that you encounter points. But it is also like you would collect clusters more perceptively than if your R value was lower. This is a very qualitative explanation of the K function. The takeaway from these two panels is that NPCs are not randomly distributed. They tend to cluster. And then this panel is, I think lamin B1 is just a protein that makes up the nuclear membrane, or like the scaffolding that's directly below the nuclear membrane. They want to see if lamin B1 interacts with NPCs at all. And their conclusion on this merged image is that NPCs are usually like existing in the gaps of lamin B1 and yeah, that makes sense because NPCs are like a hole. Like you would need scaffolding to support those holes, but the scaffolding would not be directly on those holes. Um, I think a something that they are interested in is I don't remember what they call it. Let me find it. The typical length scale. This is the preferred distance that NPCs seem to have with each other. And I could be completely wrong here. This is like the shakiest argument that I have of this entire paper, but I feel like this point at the lowest part of Y
is that preferred length, and they calculated that preferred length as 400 nanometers. And they say that that number could have something to do with the thickness of lamin B1, because that's the thing that's limiting nuclear pore complexes from being closer together. So good data. I think they also said that this data like uh agrees with some other research group. So good data. And then another thing they calculated is the nearest neighbor distance, NND, which, as the name suggests, is the distance between a point and its closest neighbor. Again, they compared the experimental results with the simulation's projection because they couldn't afford to do a three-dimensional thingy. Um, and then as you see, the NND of our experiments has significant differences from if they were just completely randomly distributed, which also supports the argument that NPCs cluster. They're not just randomly distributed. Okay, I think that's the entire graph. And then my only conclusion here is that NPCs repel each other. And then in this section, the authors also did the like what would happen if you take out a few points thingy. Which again, beautiful. Like I did, this is my annotation. Good. Like absolutely beautiful logic. Mad respect to you all. But that's that of figure two. Now we move on to figure three. Let's see if I can remember. I think figure three is when they started questioning like curvature of it all. Um, so this is just the nucleus again, with the color coding corresponding to NPC surface density. And then they cared about the actin indentation, like the thing that's compressing the nucleus, as well as the regions of heterochromatin. I think they, I definitely have conclusions highlighted here. Okay, their first conclusion is that indented areas have lower NPC density, which I think is something that they and a previous research group disagreed on. And their justification for why they disagreed is that with their super resolution, like high quality imaging techniques, they can actually like image the inside of the indent instead of projecting the entire thing onto the nuclear surface, which is why they had a lot more area to work with, which means that the same amount of NPCs generate lower NPC density instead of like higher, because that would be projecting the entire crevice onto like the top, if that makes sense. And that's the indent. And then they also found that areas of heterochromatin have lower NPC values, which makes sense given their hypothesis that NPCs play a role in transcription, and that's why they have a preferential clustering. Anyway, um, okay. Is this figure four? Okay, and that's the extent of figure three. Yep. Okay. Figure four. This is where they care about curvature, I think. This is again, the nucleus, but the color code here is oh my gosh, what is this? This is intrinsic curvature K, also known as Gaussian curvature. And then this graph here is extrinsic curvature, also known as, oh, mean curvature, I think. Yes, H. Okay. I don't really, no, I do remember the difference between these two curvatures. Gaussian curvature is not as important to us here as mean curvature. I could go into it. Do I want to? Uh, let's not go into Gaussian curvature. Basically, the difference between Gaussian and mean curvature is that one of them is intrinsic, which is property that you can like detect on a like minuscule. If you were an ant on a sphere, like if you were an ant on Earth, and if you were a really smart ant with like a ruler and everything, you would be able to detect the intrinsic curvatures, but you would not be able to detect the extrinsic curvatures because it is more extrinsic. Anyway, I am so tempted to just say, let's not talk about Gaussian curvature because it didn't really give us anything. But we do need to talk about the mean curvature. The intuition of mean curvature is that the closer that H is to zero, the more the surface between a curve is like minimalized. So if you think of like when you were playing with bubbles as a kid, you take your little thing from the bubble potion, and then there's a metallic ring, and then in the middle is like the film of the bubble through which you blow through. Um, that film wants to take up as little space as possible in 3 dimensions. So it is like thin, straight here. It didn't bulge or anything like that. You had to blow it in order for it to bulge. Same idea here. Like, uh, how do I explain this? Basically, their conclusion is that the mean curvature of this nucleus is not zero, it's slightly negative, which means that it is slightly bulging outwards. It is not like a thin film. This is a terrible explanation. I don't like this explanation at all, but I can't find a better way to explain it. Okay, we're gonna let it go, unfortunately. Where is... Okay. Okay, so their conclusions is that the relationship between MPC surface density and Gaussian curvature exhibits a slightly asymmetric bell curve centered on k equals 0. We expect it to be centered on k equals 0 because it is Gaussian curvature. Slightly asymmetric, we don't know why, and the authors didn't attempt to explain it, so I guess we're gonna let it slide. Um, and then what happened here? And then, oh, where did their mean curvature thingy go? I don't know. Okay. I don't know where their mean curvature thingy is, but the next thing that they did they have a graph for this or no? Okay. I think that's all of their graphs. So the only thing that's left from this paper is just the generalized linear mixed model, GLMM, which is where they were like off the top of my head, I can think of 3 factors that contribute to NPC density, the NPC distribution. One of them is bending energy associated with like membrane curvature. Um, this is from a theory that cell membranes have a preferred curvature, and if you bend it beyond that point in either direction, it's going to cost you energy, and that's the bending energy. Um, so they theorize that bending energy and therefore curvature is correlated to whether NPCs want to be on that surface or not, which is a fair hypothesis. The second factor is the proportion of overlap of the bin with an indentation. This is the actin indentation that we kept talking about. Again, fair. And then the third is the proportion of overlap of the bin with heterochromatin. This is the heterochromatin areas that we were talking about. Fair. So they were like, these are the 3 independent variables that we can think of. How much do each of these variables impact NPC distribution, and do they cross over at all? Like, is one of them an artifact of another that didn't give us many new information? Like, how do these 3 factors interact with each other? So they basically ran a regression model of like an equation with all of these factors um like summed together. And what they found out is all of these independently have an impact on NPC density and distribution, also known as like a region with the same curvature, but one of them is on an indentation and the other is not. They would show different NPC density distribution on the region on if both regions are in the indentation and but they have different curvatures, they would also show different NPC distribution. Like all of these factors are legit and independent of each other. That's not saying that they don't interact with each other at all. It's just that they don't completely overlap. So our model like needs all of these inputs. And I think that's it. They built a model of okay, let's talk about the model for a little bit. They built a model of NPC preferred curvature. This is directly from statistical mechanics, and I am all here for it. Basically, the idea the model started by saying, what is the energy cost of hypothetically inserting one NPC into this collection of NPCs? And they said that is this energy right here. 1/2 B is the stiffness spring constant, whatever you call it, which is great because we care about nuclear stiffness, and stiffness shows up in this model as like a legit factor. And then H is our mean curvature, and then C0 is like the membrane preferred curvature. This is like Hooke loss, like the spring constant thingy that you might see usually written like 1/2 kx squared. It's like basically the same idea, but we're using curvature instead of displacement, and we're using stiffness instead of a spring constant. And then like any large assembly of things, we want to model it with the Boltzmann distribution, which is this equation right here. Um, and then if you substitute energy inside, it looks like this. And then this is just a great bell curve, which is the bell curve that we used in this graph to model the data. So if you look closely at how Gaussian distributions work, you would find that the preferred curvature of C from our experiments is roughly equal to negative 0.25 micrometer inverted. I don't actually know how to pronounce this. Like I've been writing this, I just don't know how to pronounce it. Anyway, um, and then you also have a local radius of curvature. This is because of how they have decided to calculate curvature. Not really important, but it is quite interesting that they found like if all of their reasoning is like legit and all of their calculations and modeling are correct, then the cell membrane has a preferred curvature of negative 0.25. And that's a very interesting result. They also mentioned how this is the curvature that biologists have measured in cells at the beginning of their cell fates, like when the cell was newborn and is still growing instead of spreading, which makes sense because spreading is like I think they called it like the illness stage of the cell. It's not a healthy cell anymore if it's spreading. So it's interesting to know that when a cell is in its healthiest state, it is resting in the preferred curvature of nucleus. Anyway, I think that's it. That's the paper. Their discussion session basically summarizes everything we have done so far. They have a very detailed materials and methods section, a lot of equations on how to calculate various curvatures that my wonderful differential geometry class already told me. Um, very excited to see so much math in a biology paper. I mean, this is biophysics, so I didn't expect anything less. But yeah, this is the rest of the paper. Um, yeah, and I can't find anything else that I wanted to write about. Okay, that's great. Um, let me show you the title again if you want to read this paper in your own time. Um, General feedback for this paper is that it is amazing, and I am so, so, so excited that it exists and that I have read it. Again, mad respect for how much effort they put into checking their own logic, making sure that their methods are like experimentally relevant, physiologically relevant, and that even if mistakes or like uncertainties existed in this experiment, it didn't impact their results in a significant way. That is definitely something that I don't see often in like papers that I read. Um, I mean, I'm an engineer, so not to hate on my own species, but we are not very good at error analysis. But this paper does it beautifully. Um, yeah. Um, I will try very hard to do this again. Um, I would also consider just like talking to my camera without like any sort of presentation ahead of me. That sounds fun. Um, anyway, thank you for watching. If you're watching, I doubt anyone would watch until this point. Yeah.